Welcome to the Beer Taster's Notebook, the show dedicated to learning about beer. I'm Jonathan Thompson, advanced cicerone, beer judge, home brewer, and beer writer. Welcome to the Beer Taster's Notebook, Episode 3, How We Taste. Or, more accurately, how we perceive flavor, not how we actually taste. So I think it's a pretty good idea in the world of beer tasting to really understand how we taste. You know, quite often you hear people say things like, "Oh, I have different taste buds," or "my taste buds are off today." But what do they really mean? I mean, probably not what they think they mean. So taste is really defined as one of the five established, tastes, incidentally: sweet, sour, salty, bitter, and umami. And umami is really defined as that savory taste, sort of savory or meaty, like a mushroom. It was discovered in 1908 in Japan and really only was recognized in the West in the early 2000s. Now, there's a lot of work being done on perhaps other tastes. There's a lot of evidence that there are some fat receptors, calcium receptors, and then perhaps some receptors that detect certain metal ions. But predominantly, we're talking about the established five. And so what ends up happening is there's a chemical reaction with the nerve endings on the taste buds. And the taste buds obviously are predominantly associated with the tongue, but they do exist throughout the body as well. And we'll discuss that a bit later. And so when we say something tastes like chocolate, what we really mean is the flavor, right? So, of course, no one's going to say that flavor's like chocolate because it sounds stupid. Although with the evolution of language, I mean, maybe in 50 years, that's what we'll be saying. Who knows? So taste is, of course, an important part of flavor. It's not the main player. It's probably the secondary player. Sight, sound, and touch all play a role as well. They're probably the tertiary players, but the primary player is really aroma. And so the summary of this episode is that the brain takes inputs from the sensory system, sight, sound, texture, taste, and aroma, and assembles them all into flavor. And so there's a pretty famous, jelly bean taste test out there. It would work with everything, but I think a jelly bean is just accessible and you can just eat one, right? So basically what you do is you pinch your nose, you put the jelly bean in your mouth, you keep pinching your nose the whole time, you chew it and you see, what you're getting from it. So depending on the flavor of jelly bean, presumably you're going to be getting a sweet flavor. I don't know, maybe there's salted jelly beans, maybe there are bitter jelly beans, maybe like that Harry Potter set or something like that. But predominantly you're just getting sweet. But you would never be able to say, well, this is pear or this is cherry or this is strawberry because you're not getting any of the aromas yet. Then you unpinch your nose and a rush of air is going to come out through the back of the nose. It's called the retronasal pathway. And then all of a sudden the flavor is immediately identifiable, assuming you, of course, know what that flavor is. And so retronasal really is the biggest contributor to flavor. So if you think about the shape of most mammals' faces, humans and primates really, but are uniquely positioned that we have these shorter snouts so that the distance between the back of the throat and the nose is shortened and it's a more open pathway. So in the case of a dog, they've got this longer snout with the olfactory receptors just sort of up in there. And so they can sniff from the outside and they're getting all the orthonasal smells, but what they're eating is just kind of going straight down and they're not getting that retronasal smell as far as we know. So the brain takes signals from all these different pathways and then assembles it in the orbital frontal cortex as a flavor image. So let's think about why we actually can taste. So the core point of really any organism is survival, just long enough to procreate and ensure the survival of those offspring so we can pass down genetic material. And part of staying alive is locating and ingesting nutrition. Equally important is avoiding death. And so sight and sound provide some pretty good cues from a distance. I mean, the roar of a saber-toothed cat or the bright glow of a ripe mango indicating some nutrition. In fact, the sight of a saber-toothed cat, for that matter, is probably a pretty good signal of danger as well. And then getting closer into the body itself, pain and temperature receptors let us know of potential harm and of course some pleasures. I mean, if you put your hand on a hot stove, you'll immediately retract your hand to avoid any danger or any harm. But there's the pleasure of sliding into a warm bath when it's a cold day. And then, getting even closer now, we reach, taste and smell. So just sticking with the fruits, ripe fruits throw off esters, indicating that they're ready to eat. Rotting corpses, will throw off offensive amines, letting us know that maybe this is not something we should be eating. The taste receptors in the mouth will give us instant feedback as to the ingested material constituents as well. The fuel of life is glucose, and we're programmed to find it. There's some work suggests that umami receptors are built to identify a range of amino acids, which are, of course, the building blocks of all the essential proteins that we need. And salt is required for various functions, including nerve impulses and fluid balance, while sour and bitter are warning us of potentially harmful or spoiled compounds. So let's dive into how all this works. And let's start with the mouth. So if you remember the old tongue map that you probably learned in school, sweet was on the tip, salty was sort of flanking the tip, umami was in the middle, sour was on the sides towards the back, and then bitter ran across the back. So this was popularized by a man named Edward Boring. He kind of misinterpreted some information, and then that perpetuated for far too long. So this is a myth. It's false. There are certain areas of the tongue that may have a higher concentration of specific receptors, but the whole tongue is covered in taste buds, and all the taste buds have all 5 receptors. So there's no zone for sweet and salty and sour and such things. Strangely enough, I did a wine pairing course not too long ago at an established American university where they were still pushing the old tongue map as gospel. And so, as we mentioned before, we have 5 established tastes, a few more that are sort of coming into play. As we mentioned, fat, calcium, certain metallic ions, perhaps some complex carbohydrate receptors. And there are some studies that have shown evidence that one of the receptors that detects sour, OTOP1, also responds to ammonium chloride. So just a side note, if you've ever had Danish licorice, that's what that is. If you haven't, go and get some. It is a pretty wild experience. So on the surface of the tongue, the taste buds are embedded in the papillae, there are 3 types. Well, there are actually 4, but only 3 of them host the taste buds. So the fungiform papillae are like a mushroom shape. The foliate papillae are fold-like shapes, and the circumvallate papillae are large and round, and they reside towards the back of the tongue. So each taste bud has a pore-like opening into which the tastant molecules go. There are sort of hair-like microvilli that sweep the molecules into the receptors, and each bud has about 50 to 100 receptors and contains all 5 different types. And so when these receptors are activated, a signal is sent to the brain. The receptors interact with one another, and then with nerve endings of 3 nerve cell types, namely the 7th, 9th, and 10th cranial nerves. So the 7th nerve is the facial nerve, which is also responsible for facial muscles. The 9th is the glossopharyngeal nerve, which is involved in swallowing and saliva regulation. And the 10th is the vagus nerve, which is the main nerve of the parasympathetic nervous system. And that hosts a bunch of responsibilities all the way from digestion to mood. So the taste buds don't actually have their own axons, and they're also much less sensitive than a lot of other receptors in the human body. This makes sense when you think about that constant onslaught of stimulus that they're exposed to. And so there's a fair amount of conflicting information as to cell types, 1, 2, 3, 4, and basal cells versus 1 and 2 versus what's salty is 1 and sour is 2. So the basal cells are cells that then grow into other receptor cells. The cells that detect sweet and bitter are sort of the same type for they're GPCRs. So for the sweet receptors, there are 2 subtypes, T1R2 and T1R3. So GPCR, they're G protein coupled receptors. It's kind of like an on-off switch. And so when sugars are detected by these receptors, they activate a secondary messenger system with cyclic AMP, which causes the release of some ATP, which then binds to a type 3 receptor, which triggers a serotonin release and then stimulates a bunch of different neurons. And these receptors also detect umami via links to second messengers, the carboxylate anion of L-glutamate. The bitterness receptors behave in much the same way. The bittering compounds bind to one of the 35 known subtypes. which are, by the way, coded by more than 100 genes, just showing you how important bitterness actually is, or the detection of bitterness to keep us alive. And this is also a G protein coupled receptor, which then sends sort of a cascade of signals, which then culminate in the brain. Sourness gets a little easier. So acids contain a plethora of hydrogen ions, which flow into the receptor, sending a signal to the brain. And then salt, it's actually even easier. It's a membrane channel, and it allows potassium or sodium ions to flow through, which also trigger a signal to the brain. So, of course, the reasons for all these receptors, as we've discussed, is to keep us alive. Glucose being a readily available form of energy, and the basis of all energy on Earth, really, all plant life. And so these sweet receptors send signals to the brain that this is something good and that we need this. The fat behaves in much the same way. Unfortunately, human evolution is a bit too slow to keep up with the sudden abundance of accessible sugars and fats. And there's sugar in bottled salad dressing. Like, just consider that. So obviously, this is causing us a few issues as a society in general. And we also need salt to regulate quite a few bodily functions, and so the ability to detect salt is really also key to staying alive. Obviously, too much salt is deadly, and so this is why a little bit of salt on something tastes delicious, but a spoonful of salt would be quite off-putting. It also does seem that the brain is quite good at controlling how sensitive these receptors are based on what our current balance of salts in the body is. And so when the body is depleted of salt, it tends to crave salty things a bit more, and they taste quite good. But then things will taste even more salty than they normally would if your salt stores are quite full. And as mentioned, sour and bitterness are warnings. Souring is at times a signal that something is spoiled, although go tell that to a fresh lemon. And bitterness is a plant's defense system, and really it's associated with some of the deadliest alkaloids on the planet. Incidentally, some of the best foods for us are bitter, and liking it can be a bit of an acquired skill. If you think about the first cup of coffee you ever had, or maybe the first quadruple IPA, since we're talking about beer. But it also seems that differences in genetic makeup result in different levels of either admiration or aversion to bitterness. So there is a group of people that make up about 25% of the population, and they're known as super tasters. The term is a bit misleading, but it's not people who can, it's not a bunch of wine experts who can go, oh, this is notes of blackberry and barnyard. It's incidentally the sensitivity to bitterness. And so there's a variation in the bitter receptor gene TAS2R38, which seems to be the culprit, and that encodes a receptor on the tongue. There is still a lot of controversy out there as to causality, so we'll just sort of leave it there. But either way, super tasters react much more dramatically to bitterness and then tend to harbor an elevated affection for sweeter things. And so umami, as mentioned, is sort of defined as L-glutamate, which is a protein. It doesn't seem to trigger much reaction on its own, but when it's combined with other basic tastes, it sort of elevates everything. And a good way to think about this is MSG, which is monosodium glutamate, and that really does boost the flavor of pretty much anything that it's on. So while we're still in the oral cavity, let's talk about irritants. The capsaicin, which is chili heat, is probably the classic. So there's a receptor called transient receptor potential cation channel subfamily V member 1, also known as TRPV1, and this is part of the trigeminal nervous system. It is an ion channel. And so the main function is to detect harm, primarily temperature, and it's triggered by heat over 43 degrees centigrade. So capsaicin is an amide, a member of the vanilloid family, meaning that it has a vanillyl functional group. We don't really need to get right into the weeds on that, but it behaves in a way that it binds to the receptor and essentially lowers its threshold, which depolarizes the nerve and allows it to send signals to the brain. So basically, it tricks your body into thinking that something is really hot, even though it's not, temperature hot. So these receptors are prevalent in at least most mammals. Birds, incidentally, do not have this reaction, and which allows them to sort of spread the seeds around. just as a side note, it is thought that the plant started developing capsaicin as a deterrent, not even just a deterrent, but basically an antifungal to stop certain funguses from infecting the peppers. And so, of course, these receptors are not isolated to the mouth. Anyone who's handled a hot pepper and then touched their eyes, or perhaps just visited the toilet the next day after a very spicy meal, should be very aware of this. And so, just continuing on the same path, menthol is a monoterpenoid developed by specific plants, and it triggers the TRPM8 receptors. And then it gives them the same sort of feeling as cooling. I mean, these are also temperature receptors that would detect coolness, and so it's now tricking them into thinking that things are cooler than they actually are. Some other irritants like horseradish, mustard, wasabi, they trigger the TRPA1 receptors, which are actually in the nose. And we'll just use one as an example. So when horseradish is cut, the cells get damaged. The enzyme myrosinase is released and reacts with the synegrin, which is then hydrolyzed into allyl isothiocyanate, AITC, and that's responsible for the pungency, also of other crucifers like broccoli and Brussels sprouts. So one of the coolest ones out there, I think, is the Szechuan peppercorn. So the active ingredient there is it's hydroxy-alpha sansual, and the reaction is not really entirely understood yet, but it does stimulate TRPV1 and TRPA1 channels. It's also thought that it inhibits certain members of the tandem pore domain potassium channels. And so what ends up happening is there's this numbing, tingling effect. The cool part is that numbing, tingling is the same sensation as if something were vibrating at 50 hertz. I mean, at least I think that's pretty cool. So now moving to the somatosensory system, which refers to touch and temperature, pressure and pain. The temperature we've already sort of discussed, but the pain-sensitive terminals come from very small nerve fibers, and the TRP channels are still involved to some degree. These can be stimulated mechanically, chemically, or of course via temperature. And the touch-sensitive terminals are faster, medium-sized nerve fibers and can sense very small depressions, roughness, and any stretching of skin or membrane. TRP is again involved here. And so pressure-sensitive terminals called Pacinian corpuscles are very sensitive to vibrations. In the nose, certain receptors react to certain volatile chemicals as well, like ammonia. If you think about smelling salts, carbonic acid, which, I mean, if you take a big couple of gulps of beer, especially Belgian beers that are highly carbonated, and then you kind of burp and it comes up through your nose, that little burn there. And all of these receptors are on tips of fibers belonging to the trigeminal nerve. And so impulses travel to the trigeminal ganglion and then onto the trigeminal nucleus in the brainstem, and then output cells send those axons to somatosensory nucleus in the thalamus, and then those are relayed to the somatosensory area of the cerebral cortex. Okay, so now on to the big guns, aroma. So human beings have around 400 aroma receptors, and there are over 1 million unique aroma molecules out there. And so we probably are going to need to do an entire episode just on aroma, but you should know that aroma molecules are basically volatile organics, and they differ in a few ways. So either the chain length, the side chains and functional groups, the positions of those functional groups, chirility, meaning the left to right orientation, and then the shape, like is it a ring or is it a chain? And these all obviously affect the type of aroma that we're able to detect. So the aroma receptors reside in the olfactory epithelium, which is a 5 square centimeter bit of tissue about 7 centimeters up inside the nose. Each receptor has a bunch of tiny little cilia, which are kind of like hairs that stick out from the receptors, from the receptors' dendrites, rather. And the dendrite is like kind of like a branched extension of a nerve cell, just so you know. So the cilia are covered in olfactory receptors. And these olfactory receptors are also G protein coupled receptors, which we discussed earlier with certain taste receptors. So the G proteins, guanine nucleotide binding proteins, is what they're actually called, if you really wanted to know. Anyways, this type of signaling protein. So basically it's an on-off switch, as I mentioned. They bind to external molecules and they turn on. The process is not prohibitively complex, but it's probably not extremely important for this discussion. So they form a kind of matrix in the mucus in the nose. And each receptor has only one type of olfactory receptor. But many of the receptors have the same types that bind to many of the same aroma molecules. And they're all organized and so that all the receptors sharing receptor types have axons that converge into the glomuli in the olfactory bulb. So hopefully that makes sense. But basically, when stimulated, these receptors send signals to the olfactory bulb, which is at the base of the forebrain. And it's shaped kind of like a bulb, hence the name. And that's where we sort of begin the flavor journey. Now, there's around 1,000 genes in the human genome, specifically for OR receptor types. And so back to the brain here. So the glomerulus is the signal meeting room, if you want to call it that, right? And the receptor cells, axons, converge here and then connect with dendrites in the brain. Thousands of signals from multiple receptors show up here, and it's really the key to filtering out signal to noise. So if you think about like when you're sitting in a really loud room or bar, probably if you're a beer taster, and someone's trying to talk to you, I mean, you have to signal or you have to filter out the signal to noise, and the signal being the person that you'd like to speak to, and the noise being all the people who you don't want to hear whatsoever on the TV and the music and whatever else is going on. So output fibers from the olfactory bulb converge in the lateral olfactory tract, which then connects to the olfactory cortex. And this is where the external stimulus meets internal perception, and then the odor object is created. So part of the formation of this odor object is comparing it to the resident sort of, if you want to call it odor database, right? Like smells that we're familiar with. So, you go and smell cinnamon. I mean, you've smelled cinnamon before, so you go, that's cinnamon. If you'd never smelled it before, all of a sudden you get novel stimulus and your body has to, or your brain rather, has to sort of now paint new pictures and start reloading the database or loading a new slot in the database, if you want to call it that. So we should probably note that constant stimulus can reduce sensitivity. So, it's kind of like how you don't really know the smell of your own house, right? Or if you actually, a better example, go to someone else's house and they haven't cleaned the litter box in a while, it's going to smell pretty bad. But then, you sit around and you have a couple of drinks and some conversation and maybe play some Trivial Pursuit, and eventually you'll just get desensitized to the disgusting smell of their litter box. And this is exactly why beer judges or beer tasters would sniff their sleeve or the inside of their elbow or something just to sort of resensitize themselves. And so sensitivity also seems to be regulated by how hungry we actually are. So when we have a need for nutrients in food, we're going to be more sensitive to different aromas because we're out there looking for, I guess it's food-specific aromas, but we're out there looking for them, whereas when we don't really need a cinnamon bun, we don't have the need to be super sensitive to it. And so something else where the olfactory system stands out is where taste signals have to travel through the brainstem and the hypothalamus first. The human olfactory bulb is a single synapse away from the neocortex. So it's instant. I mean, it's probably the quickest pathway of all the senses to where we need to be processing things. And so the interesting thing about aroma also is that with all of this arsenal, we're still incredibly bad at translating the signals into usable language. And it's much like faces. So you can walk once again into the same noisy bar and immediately recognize your friends and pretty much anyone with whom you're acquainted. So, I mean, if you've met someone a couple of times, you're probably going to remember their face or at least recognize it. Okay. So now picture one of those faces and then describe it so that someone else can form the same image. It's really not so easy. I mean, if you take a look at some police sketch artist work, I mean, they're highly trained, but they're still very strange looking pictures. And this is just a result of just, I mean, it is just really hard. First of all, I mean, obviously there's some other issues at play like misremembering, et cetera. But I mean, it's just very, very hard to describe faces, just like it's hard to describe aromas. And so let's move into some of the tertiary stuff for a bit. So sounds, auditory stimulation of food is likely something that's learned. the nutritional value of a soggy carrot versus a crispy one is likely going to be the same. Sour dill pickle that wasn't prepared in a proper way is going to be really mushy and kind of off-putting, but it's the same thing. It's still a cucumber and it's still covered in lactobacillus and salt, but it doesn't. It's gross. And this doesn't make. Just because it's still nutritionally viable doesn't mean it's the sound is any less important. So when that snap of a carrot kind of, it tells you that's what you want. And when you eat potato chips, you want that crunch and you want to hear that crunch. It's not just the vibrations or it's not just the sensory in your mouth. You really want to hear that, or the glug of wine. You wouldn't want to drink silent wine and you wouldn't want to eat soggy or silent potato chips, the texture aside. And so these all contribute to part of the experience. And, when you think about the mental picture that you're drawing as well, if you open up a can or a bottle of beer and there's no psh sound, you're immediately forming preliminary images of a flat beverage that's not really something that you're after. And so when it comes to sight, there's always these things that people say, we eat with our eyes, and visual stimulation plays a huge role in flavor. So just riffing off this potato chip theme that I seem to be obsessed with, a really dark brown or maybe like a black potato chip, and this is some funny flavor, like it's charcoal flavor or something like that. It's going to be kind of, it's not going to be very appealing. You want your potato chip to look like what you want it to look like. And you might have, images in your mind of it being burnt or moldy or off. Or maybe think about something a little less gross, like a chip that's coated in red powder, but it tastes like sour cream and onion. You expect it to be a ketchup chip. And so we are sort of predetermining what things may or may not taste like based on how they look. There's a lot of work out there on plate sizes and plate colors. And if you put popcorn in a blue bowl that tastes saltier, I believe is the one. There's a famous study where they gave a bunch of wine experts, and I think that they and everyone else reporting on this always likes to emphasize that these are expert tasters. So they give them various wines, and they obviously have to describe them, and they serve white wines and red wines side by side, and then they serve the same white wine, but they dye one version red. Other than that, it's exactly the same thing. And so these tasters, these experts, are all kind of fooled, and they start finding red wine qualities in the whites. So their brains trick them. It's not their fault. Well, I guess it is because it's their brains. But, this is just all part of the experience. And after all, it is all in your head. And so, in the case of beer, which is really where we're concerned, if you're more liable to taste roasted notes in a beer that's black, regardless of whether it's there or not. And then the Coke versus Pepsi. I don't know if you remember that challenge. That's another example of the effects of visual stimulation, or in this case, the lack of. I never really understood that one because I feel that there is a distinct difference in flavor between the two. And it's a silly debate anyways, because clearly beer is better than both. So hopefully that was clear enough. It is fairly complex, and it's probably easier to read through these things and look at diagrams. But now let's put it all together. And I guess we can use food only because I do want to talk a little bit about chewing. And yeah, sure, I'll use the potato chip. So you see the chip in your hand, right? And you can feel it between your fingers. So of course you're already stimulating the touch. The visual stimulation signals are already there, preparing the canvas. You see the color of it. You see that it's, and it looks like kind of off-white with some green flecks. So you kind of have an idea that it's sour cream and onion. You probably know because you purchased it, but either way. And as it gets closer to the face, we start to be able to smell it. That's orthonasal, the ingesting of aroma molecules through the nose. We can smell that it's sour cream and onion. And now we introduce it into our mouth. And as we bite down, there's a crunch. We hear it. We feel it. Those signals are firing. All of a sudden, the salt receptors on the tongue are firing. It's sour cream and onion, so there's a little bit of sweetness in there too, and that's firing. And now the shards of broken chip are stimulating pressure receptors through the mouth as we continue to chew. The fat in the chip is sending strong signals as well. And the chewing is breaking the food down, releasing aromas, which are detected retronasally. And that's probably one of the most important parts of flavor. Swallowing forces the air up into the nose, which takes with it a bunch of volatiles and picks up even more that are sort of stuck into the palate, the sort of aftertaste compounds. The brain is going to grab all these signals, reference them, and then produce the flavor image. So I'm going to go slightly off course here just for a second to talk about saliva. It'd be really hard to sort of integrate the role of saliva in without losing the flow, but it is quite important and very interesting. So saliva is 99.5% water, and it serves to dilute food molecules and then flood the taste buds with them. Mucins are proteins that bind to aroma compounds, moderating the release and then contributing to the aftertaste as well. These are primarily MUC5B and MUC7. Also, they only make up about 0.3% by weight of the saliva, but they do carry a big stick. There are also a ton of salivary enzymes, but the ones that are most important are superoxide dismutase, which catalyzes the redox reactions, cysteine B lyase, which is produced by oral bacteria, and it actually breaks down S-methyl L-cysteine sulfoxides in vegetables, which produce those sulfur compounds. So when you bite an onion or a clove of garlic, those enzymes are responsible for that pungency. So those are pretty cool, but the really big players are lipase that begins fat digestion. It's far less active in the mouth, incidentally. It really does prefer a pH range of about 3.5 to 6, and so it gets quite busy in the stomach. And then the most important of them all, the amylase enzymes. So brewers of all types will know all about amylase enzymes from the mash. But it is the most abundant enzyme in saliva, and its function is to break down carbohydrates or starches into smaller sugars. And this, of course, contributes to taste. And a cool little side note on the power of salivary amylase. So if you make a pot of oatmeal, split it into 2 bowls, eat some from one bowl, and eat it normally. So the spoon's going into your mouth and then back into the bowl, of course. But leave the other one completely untouched. And then put them in the fridge or leave them out on the counter or whatever, but just make sure they're in the exact same environment. After some time, when you check on them, probably the end of the day, the untouched one will be that congealed goop of what old oatmeal is, and the other one will be kind of watery and separated. So just the amount of amylase that has made it into the bowl from the spoon, and remember that it doesn't take a lot because they're not consumed in the reaction. And so through the day, they just are busy just chopping up starches. And so you've got pre-digested oatmeal now in the bowl on the counter. So I'm sure that's really appealing to everybody. And a final interesting note on saliva is it does seem that certain esters are hydrolyzed in the saliva, and that actually reduces their retronasal stimulus potential. Just a few interesting points about different sensory interactions before we sort of wrap it all up. So temperature can affect both texture and irritation. Like if you have spicy food that's sort of, room temperature versus spicy food that's quite hot, it's going to elevate that irritation. Hotter foods also can taste stronger, both because of the increased volatilization and also, the reduced viscosity, which allows more distribution throughout the mouth. And so increased viscosity does reduce some retronasal stimulation. So if something's obviously much, much thicker, you're not going to be releasing so many volatiles up the back of the nose. Sensitivity generally is the highest and the most regulated at normal mouth temperatures, though, of 22 to 37 degrees Celsius. Some irritants can increase the perceptions of heating and cooling, depending on what they are. And importantly, in beer especially, sweetness can increase the perception of viscosity. So when we think about the heavier, thicker beers, they tend to be sort of under-attenuated. So it's the sweetness that's giving us that bigger body, as we like to call it. Acids, of course, can make things taste slightly less sweet. And so the carbon dioxide forms carbonic acid when it's dissolved in water, which is, of course, very important for beer. But it's a nice real-world test is a can of any kind of soda pop. So if the same temperature, the same drink, one is fresh and one's flat, there's going to be a massive difference. The flat one's going to taste abnormally sweet. All right, so let's tie this all together and tie it all back to beer. So we don't really chew the beer, and which is why we used the potato chip example instead of beer earlier, but we do move it around our mouths. So mouthfeel, body, and astringency are all quite important. The texture we get from the carbonation as well, that all contributes to the somatosensory inputs. Ethanol can trigger some interesting responses also. It can bind to both sweet and bitter receptors, and it stimulates the heat detecting receptors. So you think about drinking like cask strength whiskey, just neat. It burns, right? Because it's actually stimulating those same receptors. The ethanol molecules can also attach themselves to odor receptors. So if you think about, Scotch nosers adding a bit of water to the Scotch, or actually cocktails are probably a better example because when they're shaken or stirred with ice, there's some dilution there that it helps reduce the amount of ethanol, so that it stops it from overwhelming all the other aromas that are otherwise nuanced and probably actually just completely obliterated if you have something that's super strong. And the cooling down also reduces some of the volatility a little bit, so you don't have all these alcohol molecules just firing around willy-nilly. So let's just look at the beer and all the visual signals and start at the top, down signaling. So I think it's a pretty rare taster who doesn't develop some kind of preconception based on the color. I mean, no one really expects a pale straw, sparkling, effervescent, clear beer to taste like chocolate, toffee, and coffee. Nor do they expect a jet black pint with a tanned, creamy head to taste like pineapple and bread dough. So we're going to be getting a preconceived notion of what we want or what we expect some of the tastes or flavors, I should be saying, to be just based on the visuals. And so orthonasal comes next, which, allows us to really start developing an image. And we smell our beers first and start to parse out whatever flavors we're getting from fermentation or from the hops or from the malt, and obviously to a certain degree from the water. And then we introduce it to the mouth. We certainly don't expect umami in our beers, but we do expect sweet, sour, bitter, and in some cases salt, like a gosa, or if there's using a water profile that's got a little bit more salt in it. The texture of the carbonation, the weight of the beer, in other words, how much the sugars are and how it feels on the tongue. So an Eisbock obviously is going to be vastly different than a Berliner Weiss for two reasons: carbonation and mouthfeel, body, I should say. And then these all come together with retronasal, both on the introduction into the mouth and on the swallowing. Sound is probably the most minor player with beer. That slight crackle of carbon dioxide maybe in the mouth, or the gulp when you swallow it, but there's not any snap or crunch as we chew it. And I mean, well, if that happens, maybe consider sending it back. And so that's our experience, but what's going on in the nervous system? So the visual photoreceptors are stimulated. Optic nerve takes the signal to the thalamus and then to the visual cortex in the occipital lobe. The olfactory nerves are stimulated. They project to the olfactory bulb and then the olfactory cortex in the temporal lobe. The taste travels through the cranial nerve to the medulla oblongata and then the thalamus and then the gustatory cortex, which is in the parietal lobe. The texture signals are sent to the brainstem and then the thalamus and then the somatosensory cortex. And with sound, the cochlear nerve sends the signal to the thalamus and then the auditory cortex in the temporal lobe. Integration of all of these signals seems to happen in the insula, and then the value is assigned in the orbital frontal cortex. And all this together gives us our oatmeal stout, or our IPA, or our lager, or our Flanders Red. And so that's how we taste, or maybe should I say how we flavor. And in coming episodes, we'll discuss aromas, what they actually are, and then which compounds specifically are responsible for which aromas in beer. And as a supplement, you can read my ongoing Aroma Files series at thebeertastersnotebook.com, where we dive into each beer aroma one by one. Until next time, stay thirsty and stay curious.